Hi everyone. Today I'm going to be going over Lab 7, growth kinetics and protein content via the BCA assay. So what I want to do now is kind of take some time to set the context of the next few labs. Last week, you guys learned how to do mammalian cell culture and all the different techniques associated with that. One of the things we're trying to help you guys learn is how to properly manage your cells, and with that comes an understanding of how fast your cells are dividing so that you could seed them properly for experiments. Now we know that counting cells manually, taking pictures of them, and recording these numbers isn't exactly the most exciting work, but it's a very important skill to learn, and hopefully you guys have been getting better at it so you can go a little faster. What we're going to be doing in the next few labs is teaching you guys ways to quantify cell number without having to count. Unfortunately, in these labs, you're still going to have to count, but in the future, you won't have to, so it's kind of a win. The exact theory and all that other good stuff you can get from the pre-lab reading. So please do that. But in short, you're going to be quantifying cell number through total protein content in this particular lab. In lab 8, you're going to do it by metabolic cell activity. But because we don't have time to go over all this pre-lab reading, I'm going to ask you to do it right now. If you haven't already, just pause the video and go and read it. I'm going to move on to the procedure because I want this to not take too long. Okay, so recall from the previous week that you have a 12-well plate and that you have 3 different seeding conditions and you have 4 wells of each. In an ideal world, we would have each of these wells be an independent assay of protein content so that you could have standard deviation between yourself. Unfortunately, we don't live in an ideal world, so instead what's going to happen is you're going to take your 4 wells and you're going to turn that into 2 tubes. One of those tubes you're going to take all the way through the procedure and perform the BCA assay on, and the other one you're going to count. So what that's going to look like, if we just split this here, I'm just going to draw it for A, but these 2 wells on the left, I'm going to trypsinize and take for BCA. And these other ones I'm going to take for cell counting. Okay, hopefully this kind of makes clear what I've tried to write in the actual protocol itself. Don't forget that because you actually have 2 wells, when you're doing these assays, you need to divide by 2 in order to get average protein content or an average cell number per well. Okay, so that's the first part. And what you guys should also be doing is during this time, because there's a couple of different incubation steps, especially like right here, okay, is that you'll also be wanting to passage your flask for experiment on Wednesday. For the experiment on Wednesday, actually, you'll notice that I'm having you guys seed 4 wells of each cell density, and we're actually going to have standard deviations for yourself. So yay. In the second part of the protocol, you're going to be actually preparing the working reagent itself. This is pretty straightforward. Just keep in mind that it's a 50-to-1 ratio. One thing is, you guys don't actually need to be preparing any working reagent for the standards. Instead, we decided in the TA meeting that your TAs will be making 1 standard per section. So you only need to make enough working reagent for your 3 unknowns. Again, while it's incubating, take some time to talk to your TA about the theory of this lab and how BCA actually works. At the same time, if you guys haven't started already, I'd recommend thinking about the independent lab projects. They're coming up soon, and you should be thinking of ideas that you can discuss with your TA so they can help you. Again, please remember to get data from other groups, as this is going to be the data you'll use for your statistical analysis and the lab reports. I've included a 96-well plate here. Your TA should bring a copy of this to the actual lab period, but just in case if you have this, you can kind of mark which wells are yours. So for example, you can have like these 9 wells up here be set for the standard, and then just write in where your wells are. So for me, I just be like these 3. All right. Okay. This procedure is actually pretty simple, so that's why I didn't want to spent a lot of time on it, but hopefully this clears up some of the instructions in the protocol. I'll see you guys next time.